So hello again, everybody. Welcome back. We are now very much into the home stretch because we have two more chapters to cover. And of course, we have to cover them this week. So in this recording, we're going to be looking at chapter 16, looking a little bit about the history of the structure of the DNA molecule, how it was arrived at as a double helix, as published by Watson and Crick in 1953. And then we'll look at one of the implications of that model, the idea that DNA is replicated in a semi-conservative fashion, and then we'll look at the actual mechanics of that replication. In the last chapter, which I will post on Wednesday, we will look at the role of DNA as the origin of instructions for assembling proteins, looking specifically at the processes of transcription and translation, and what happens when nucleotide sequences change. In other words, what happens when you get a mutation. But that will be in the last chapter, chapter 17. So I will post that one on Wednesday. But for today, we are going to be looking at the structure of the DNA molecule. So when Watson and Crick published their paper in the journal Nature, and you can go and find this on the internet, it's the April edition from 1953, and you can find Watson and Crick's paper there. And it is a remarkably short paper given the implications for what they were publishing. It's a single page, mostly text, with a ribbon model diagram of the double helix to one side on the left-hand side in the bottom left-hand corner, and then the citations, the bibliography for the paper, carry on onto the next page. But the actual paper itself is that one-page paper with a single diagram. That was the Watson and Crick paper that introduced everyone to the double helix model. Now, it wasn't published in isolation because there were two following papers, one published by Rosalind Franklin, who I'll introduce you to a little bit later, who produced the key piece of data that Watson and Crick were able to utilize, a photographic image that outlined some of the basic physical features of the DNA molecule that no one had, up until the point that Franklin produced this photograph with one of her graduate students, had been able to get. And that gave Watson and Crick an advantage, and how they got that access to that information is actually one of the more unpleasant stories associated with what we talk about in this class. Unfortunately, we don't have a whole lot of time to talk about that, but I will give you a little bit of that story. But if you really want to get the full story on what went on in terms of who had access to what and how, you can find the story outlined in many different, on many different websites. So it's not in a secret or anything like that. You can find plenty of places where you can find the full story. Yeah, I've lost my train of thought now. We'll talk about Franklin and her contribution. Yeah, I've completely lost my train of thought. The train of thought left the station, and apparently I was not on it. So anyway, okay, I was talking about the papers that were published in company with the Watson and Crick's paper. So Franklin published a paper by herself, outlining her interpretation of the X-ray image, the photographic image. And then Watson, Crick, and Wilkins, Maurice Wilkins, published a paper in which they went into a little bit more detail on the double helix model. And right there, you actually start to see part of the story behind the development of this model and subsequent publication and who got credit for what and so forth. And it is part of the story, unfortunately. And as I said, I'll give you some insight into some of that story a little bit later. But those are the four major players. There is one other player that I'll mention as well, an American, very, very famous chemist by the name of Linus Pauling. He literally wrote the book on chemical bonding. So if you want to know who to blame for ionic bonding and covalent bonding and all of that you learnt back in chapter 2, that was Linus Pauling. He literally wrote the book on bonding and received the Nobel Prize for it. He also received a second Nobel Prize, in this case a Peace Prize, for his work to try and eliminate nuclear weapons. So he is one of a number of people who actually received 2 Nobel Prizes, but he received them for different areas of work. And Pauling would actually influence the direction in which the work looking at molecules of inheritance would actually move for many years because of his fame as a chemist. He was also known for his work on proteins. And for many years, people were more inclined to think of proteins as the molecule of inheritance rather than DNA, even though there was substantial evidence that should have pointed in the direction of DNA. And we'll talk a little bit about that as part of this presentation today. So those are the people, the main players in all of this. But in terms of developing the double helix model. But before we get to the double helix model, let's talk a little bit about some of those experiments that should have really given people cause to think about whether it was DNA versus protein. So just so that you know who is who, this is James Watson. This is Francis Crick. Watson was a postdoctoral fellow. That means he had received his PhD in the United States. He traveled to Europe. He had originally gone to Berlin to work at the Max Planck Institute, but had decided that he didn't want to work there and had wound up at Cambridge at the Cavendish Laboratory, where he had joined up with Francis Crick, who was at the time a theoretical chemist, which in some sense is a pretty good description of James Watson as well. They would then start working on DNA around the middle to the end of 1952, working on it rather quietly and then not so quietly, until it got to the point where the head of the Cavendish Laboratory said, stop, this is not what you're here to do. You're not making any headway. Go back and work on what you were intended to work on. Which they did for a little while. And then some more information was published that really convinced everybody that DNA was actually the molecule of inheritance. And Watson and Crick went back to the head of the laboratory and pled their case to get back onto the DNA story, and were given permission to start working on DNA, and would ultimately turn out to be these successful individuals to publish what we now recognize as the double helix model. And this is their first 2-scale model of the DNA molecule. So obviously it's substantially larger than an existing DNA molecule, but it is nonetheless built to scale. It's an extrapolated model. And you can actually see this model. It is in a glass case in the Natural Science Museum in London. I've had the privilege of seeing it. It's in the foyer. There is a second model in the new Cavendish Laboratory building, which is a few miles outside of Cambridge, in the little museum that they have there. And you can go and see that there. And I've actually been to the laboratory as well and had a chance to look in the museum. So I've seen both of the models that Watson and Crick built to try and explain what the double helix actually looks like. So this is the first one that they built. So this is Watson and Crick, just so that you know who is who, and you get an idea of some of the people that we're talking about this morning in this presentation. So let's take a look at some of those experiments that I alluded to. That should have given people some pause to think about the role of DNA. So one of the things that we have to remember in all of this is that once the work of Mendel was recognized for its significance in 1901, and you may wonder why we're talking about 1901. Well, we have to remember that this is when people started to put the connection together between Mendel's work and chromosomes. Because remember, chromosomes were known. The process of mitosis was figured out in the 1880s. The process of meiosis was figured out in the 1890s. And when Mendel's work was finally recognized and people started to think about the process of inheritance and Mendel's inheritance particles, what we call genes and alleles today, they reached the conclusion that these must be located on chromosomes. We've talked about this before when we talked about the work of Morgan and his fruit flies. Because remember, it was Morgan who finally produced the first convincing evidence for the physical location of a gene on a particular chromosome. In this case, it was the eye color gene in fruit flies that he localized to the X chromosome. And of course, the extension from that then is that all genes are on chromosomes. So right there, you have chromosomes as being the structural unit involved in inheritance. But chromosomes are not made up of a single molecule. They're actually made up of two molecules. They contain DNA and they contain protein. And here's where people had to think about which direction they were going to move in terms of investigating these two molecules as the potential molecules of inheritance. And you could kind of imagine them reasoning in this sort of a way, because by the time this decision was made, which was made in the late 1920s, early 1930s, that in fact, it was likely protein that was the more likely that protein served as the molecules of inheritance rather than DNA. It had turned out that inheritance was a far more complex process than Mendel had ever described it as, because building on the work of Mendel were people like Morgan and his graduate students and others working in the area of inheritance. And suddenly Mendel's explanation looked fairly simple compared to what people were discovering. The process of inheritance was turning out to be way more complicated. And when you compared the two molecules contained in chromosomes, DNA, which came apparently in only one form, was made up of only 4 subunits, what we now call the 4 nitrogenous bases or the 4 nucleotides. And then compare that with proteins that come in literally hundreds of thousands of different forms and were made up of at least 20 subunits, the 20 common amino acids. We know that there are a few more, but 20 common amino acids turn up commonly in proteins. It's sort of a no-brainer in terms of thinking about which of the two molecules might be involved in a very complex process, because it stands to reason if you've got a complex problem, a complex solution is likely to be at the heart of it. This is a basic idea that is encapsulated in an idea called parsimony. Parsimony says that the simplest explanation is the best explanation. And so for a simple problem, a simple explanation is probably the answer. But for something that is more complicated, the answer is probably going to be more complicated. And so when you look at inheritance and you realize that it's very, very complicated, then a complex answer is more likely to be in the form of the proteins, not so much in the DNA, because DNA is too simplistic, or at least that's probably how the argument went. And so starting in the late 1920s and on into the 1930s and 40s and early 1950s, a lot of effort was placed on trying to figure out the basic structure of proteins. And you might argue, and some people have, that this effort was largely wasted if, in fact, your idea was to figure out the basis of inheritance. as it would turn out, a technique that was developed for looking at the three-dimensional structure of proteins and deciphering the three-dimensional structure of proteins, something that Linus Pauling and his group were exceptionally good at doing, and that Maurice Wilkins and very particularly Rosalind Franklin were outstanding at doing, would provide the key. And it was Franklin's ability to utilize this technique that would produce the key piece of evidence that provided the key to unlock the double helix model, as you'll see. But in the meantime, people were still investigating DNA. It was, after all, a molecule found in cells, so it was worthy of study nonetheless. And there were some experiments that were carried out at various times in the 1920s, 1930s, and 1940s that should have given people, again, pause to think about whether protein was should be considered to be the molecule of inheritance. Maybe it really was DNA. So let's talk about a couple of those key experiments. So the first one was carried out by a British doctor named Frederick Griffith. Griffith worked for the National Health System in the United Kingdom. That is the public health system that provides free medical care to all members, to all citizens of the United Kingdom and people who are resident there. And what Griffith was doing was studying the bacterium Streptococcus pneumoniae, which is the bacterium that causes pneumonia, because pneumonia was a death sentence at this time. There were no antibiotics when Griffith was doing this work. This was in the 1920s. He published his paper in 1928. And so what he was trying to do was to create a vaccine against the bacterium, because the concept of vaccination went back best part of 150 years. The person given credit for it is another British doctor by the name of Joseph Lister, who is credited with carrying out the first experiments using cowpox as a vaccine against smallpox, based on the observations of the effect of cowpox on milkmaids and their apparent resistance to smallpox. We now know that, in fact, Lister probably wasn't the first person to observe this. In fact, a dairy farmer may have been the very first person to observe this and to have actually spoken publicly about it. And it is likely that Lister heard about this and, as a medical doctor, went ahead and did this and probably had greater credibility as a consequence of this. That and the fact that the work of the dairy farmer, and I've forgotten his name right now, but I was reading an article about him just a couple of weeks ago, did not widely publish this, even though when Lister was given credit for this, some people argued that the dairy farmer should have got credit, but Lister had some very highly placed friends who got him credit and money from the royal family and so forth, and so he has been widely credited with vaccination. Anyway, I digress. So the idea of vaccination and how to create a vaccine was pretty well understood in Griffith's time. What he knew he needed was a non-pathogenic version of the Streptococcus pneumoniae bacterium that would not kill, but would trigger the immune system to produce antibodies against the bacterium. And the good news is that Streptococcus pneumoniae fits that bill precisely. There are two strains. One is a pathogenic strain, otherwise known as the S strain or smooth strain, because it produces a coat material made out of some very toxic substances called lipopolysaccharides that protect the cell from the immune system of the host, whether it be something like a rat or a mouse, because this bacterium causes pneumonia in all mammals, including humans. And then there's another strain, which is referred to as the R strain for rough strain, which does not produce this smooth coating and therefore is accessible by the immune system and therefore can be killed. It is not pathogenic. And Griffith was going to use these two strains, and he was hoping particularly to use the rough strain, to develop a technique for creating a vaccine against the pneumonia bacterium. Because if you were infected with pneumonia, it wasn't a case of if you would die, it was a case of when you died. It was a death sentence. And contracting pneumonia was one of the reasons why there was migration in the United States, at least, from the cities on the East Coast out west, because it was known that a drier, warmer climate could at least reduce the severity of pneumonia and give you an opportunity to live longer. And if you were really lucky, perhaps even cure you or put you in remission. You may never be truly cured, but you may not suffer from the ravages of pneumonia. And so it was pretty important under these circumstances that some way of protecting people against pneumonia could be developed. It really didn't come about until the development of antibiotics. The first antibiotics became available in the mid-1930s in the form of sulfur drugs, and then you get penicillin. And from that point onwards, you start to get a wider availability of antibiotics, particularly after World War II. And so pneumonia today is not considered to be quite so problematic, although we do now recognize that some strains of pneumonia are becoming resistant to the antibiotics, which is another story for another day, but it is becoming more problematic. So here's the basic technique that Griffith adopted. He started out by double checking to make sure that the strains would do in mice what he expected them to do. So he injected living pathogenic bacteria, the S strain, into the mouse, and the mouse, as expected, died. Then he took living R cells and injected those into another mouse. These are his non-pathogenic bacteria. And as he expected, the mouse lived. So his first idea to create a vaccine was to take the living S cells and apply the technique that was first developed by Louis Pasteur. Raise the temperature of the solution containing these living cells to the boiling point of water, 212 degrees Fahrenheit, 100 degrees Celsius, and hold the temperature there for several minutes, and that temperature, that heat, would kill the cells. He knew that this is how you could kill bacteria. You could heat kill the bacteria by using what we today call pasteurization, after Louis Pasteur. And so he heated these S cells and killed them. Now, having killed these cells, he expected the mouse not to be killed by the bacteria. Always a good thing if you're trying to create a vaccine. But what he was hoping was that injecting these cells into the mouse would trigger the immune system to react against them, and therefore he could use these dead S cells as part of his vaccine. Unfortunately, the mouse lived. Well, maybe fortunately the mouse lived, but unfortunately there was no immune response because it still had that coat that protected it from the immune system. So what was he to do? Well, he knew he could not inject living S cells into the mouse because the mouse was going to die. That he'd already demonstrated. Whatever he was going to inject into the mouse had to be non-pathogenic. But he now has recognized that if he kills the S cells, he's not going to get what he's looking for, an immune response. So he's thinking, well, I've got to inject something that's living, but the only thing that I can inject that's living is the R cell, because that's the non-pathogenic form. But that's the one that won't trigger pneumonia. So how about I mix together the dead S cells, because they won't kill anything, and the living R cells, because they won't kill anything. So I'm mixing together two forms of cells that won't kill anything. Therefore, the mouse should survive, and perhaps I can trigger an immune response. So he injects this into the mouse, and imagine his surprise when the mouse promptly dies. Oops. not the expected result. But what was even more surprising, perhaps, was that when he extracted some blood out of this mouse, which is where the bacteria are located, the only bacteria that he could find in there, living bacteria, were S cells. The ones that he had injected in that were, he knew, dead because he'd killed them. Yet here in the dead mouse were living S cells. Well, that explained why the mouse died, because the S cells were there. But where did they come from? The only S cells that he had injected were dead S cells. The only living cells he had injected were R cells, and those are not pathogenic. Yet the only cells that he can get out of this dead mouse are living S cells. Here's where he had his moment of insight. He recognized that those living S cells were actually some of the living R cells that had been transformed into living S cells. And he actually used the term transformed and called this process transformation. And he further had the insight that what had happened, or what must have happened, is that those living R cells had taken something up from the dead S cells that had transformed the living R cells into living S cells. Now, he went one step further, and here he kind of stuck his neck out, because he didn't really have any evidence for this, but he had a fairly good background in biochemistry. And he knew that the heating of a cell and its death was caused by the denaturing of proteins. Proteins were extremely sensitive to heating. We learned that back in chapter 5. If you heat most proteins above 45 degrees Celsius, they lose their shape, they denature. And this is what led to the death of the S cells when they were heated. And he also knew, because people had been working with DNA, that DNA was relatively heat stable. And so if it was going to be one of the two molecules that were going to survive the heating and therefore transform the cells, change their makeup, change their phenotype, he suspected that it was probably DNA. And he actually made a comment about that in his paper. Now, he had no evidence for this. He was just speculating. And he went ahead and published this paper in 1928. And again, you can find a copy of this paper on the internet if you go looking for it. When Griffith's work was transmitted across the Atlantic Ocean, when the journal in which Griffith's paper arrived in America, it came to the notice of the head of the microbiology department at Johns Hopkins University Hospital. And the head of the microbiology department was a little bit skeptical about this notion of transformation. In fact, he was inclined to think that Griffith was wrong, that whatever had happened, it wasn't the take-up of something from the dead S cells that transformed the living R cells into living S cells. And so he assigned a couple of the individuals in the microbiology lab, McCarthy and McLeod initially, to work on this and to try and find out whether Griffiths was correct or not. So the first thing they did was to repeat Griffith's work and figure out that, yes, Griffith was in fact correct, that you could transform living R cells into living S cells using the presence of dead S cells. But then they continued to play with the system, and they eventually discovered that you really didn't even need to have the S cell present, the dead S cells present. All you needed to do was to have an extract from the S cells and expose the living R cells to that extract, and whatever it was that was in that extract that could do the transformation, the R cells would pick up and get transformed into S cells. And so they followed up with this by treating solutions of these extracts with enzymes to destroy 3 different molecules. In one solution, they destroyed all the proteins. They used an enzyme called a protease, which cuts up proteins. In a second solution, they used a nuclease, and a nuclease cuts up DNA. And that meant they destroyed the DNA in that solution. In the third solution, they used another type of nuclease that cut up RNA. So in that solution, they destroyed the RNA because they could, and because, of course, DNA and RNA are related, and people were starting to recognize that DNA and RNA worked in concert as part of the flow of information, genetic information in cells. Of course, we're starting to talk the 1930s and early 1940s at this point. And so there was evidence for the role of RNA. And so they destroyed the RNA in the third solution. Then they exposed living E. coli cells in this case, because they discovered that they could do this work with E. coli cells. They didn't need to work with the pneumonia bacterium anymore. They could basically get E. coli to transform just as easily. And so they exposed E. coli to each of these solutions. So remember, one of the solutions lacks protein, one of the solutions lacks DNA, and one of the solutions lacks RNA. And it was only in the solution that lacks DNA that they could not get transformation. In other words, in the absence of DNA, you got no transformation. In the solution that lacked protein, you still got transformation. In the solution that lacked RNA, you still got transformation. You lost transformation, or the ability to transform the bacteria, when you took out the DNA. And so they published this paper suggesting that DNA was the transforming factor based on the results of their experiment. I don't recall, because it's been a while since I've read the paper, and perhaps I need to go back and look at it again, whether they suggested that DNA was actually the molecule of inheritance, but certainly the evidence suggests very strongly that that is the case, because again, transformation is all about changing a phenotype. And we know that a phenotype is a product of a genotype, and a genotype is associated with chromosomes, and chromosomes contain only DNA and RNA, DNA and protein. And so if it's DNA that's the transforming factor, it strongly suggests that DNA must be the molecule of inheritance. But of course, this work is being published in the early 1940s, right in the middle of World War II. And I guess it got lost in the shuffle because people had bigger things to think about at this point in time, other than what was the molecule of inheritance. And we also need to bear in mind that there was still this emphasis on protein because of people like Pauling and other protein chemists who were fairly convinced that it had to be protein. And so even though this data was being produced by people studying DNA and the effect of DNA, there was this strong tendency still to sort of rationalize away the DNA evidence and continue to focus on proteins. And this is not an unusual thing. It's what we do as humans. We try and rationalize away the things that we are not comfortable with or that do not match our particular ideas. And this was one of those ideas that was very strongly held by a lot of people at this point in time, because they kept arguing. And one of the arguments against the work of Avery, McCarthy, and McLeod, Avery was assigned to this project when I believe it was McLeod left. And so the work is credited jointly to Avery, McCarthy, and McLeod as the final authors of the key papers that describe this work. The people who were convinced that it was still protein basically argued that, yes, you may have used proteases to cut up the proteins, but protease doesn't completely cut up the proteins into individual amino acids, and maybe a short polypeptide is sufficient to do the transformation, particularly if you haven't completely destroyed all the proteins, which was their argument, that the proteins hadn't been completely destroyed. Again, rationalizing away evidence that probably should have given people pause to think in another direction. So what finally convinced everybody that DNA really was the molecule of inheritance? Well, that was work done by two individuals, Alfred Hershey and Marcia Chase, working at the University of Pennsylvania, if I recall correctly, working with a virus, but not just any virus, a virus called the T2 bacteriophage. A bacteriophage is simply a virus that attacks bacteria. All types of cells are susceptible to virus attack. So animal cells can get attacked by viruses, plant cells can get attacked by viruses, fungal cells can get attacked by viruses, bacterial cells can get attacked by viruses. So there are viruses that specialize in attacking different types of cells. Now, what was particularly useful about the T2 bacteriophage, and here's a picture of it. This is an actual electron microscope image of a T2 bacteriophage, so you can see it here. And this is an E. coli cell that's in the process of infecting. And what is really great about the T2 bacteriophage is that it contains the two molecules that were in question as the molecule of inheritance. Inside the head of the bacteriophage is a coiled up piece of DNA. It's genetic material. And then the rest of the bacteriophage is made up of protein. So in essence, it's a piece of DNA packaged within a protein code. So it's made up of the two molecules that were in question as the genetic material. Now, what is even more important about viruses is that a virus can't self-replicate. It has to take over an existing cell and reprogram that cell to become a virus factory. And it does so by inserting something into the cell. We now know that it's the genetic material. We also know that some viruses do use DNA, but other viruses use RNA. And so the HIV virus, I'm trying to think if the coronavirus, COVID virus is one of these as well. But I know the HIV virus is an RNA virus. And it uses an enzyme that takes that RNA molecule and makes a copy of it in the form of DNA. It reverses the process of transcription. And so the enzyme is called a reverse transcriptase, and the process is called reverse transcription. That DNA, that single-stranded DNA molecule, is then duplicated by the cell's own DNA replication machinery, and that now double-stranded DNA inserts into the host cell's genome, into its chromosomes, where it hides until it reemerges as an infective agent and starts the cell that it's sitting in, what are known as CD4 cells that are central to our human immune system, to making more HIV virus particles, which then ruptures the cell, which kills it, and they go off to attack more CD4 cells. And that's what collapses the immune system of someone who is HIV positive and leads to the onset of AIDS, acquired immune deficiency syndrome, because you lose your immune system, which makes you very susceptible to all sorts of infectious diseases and some types of cancer that otherwise your immune system might be able to eliminate. And that's what winds up killing you. It's the loss of your immune system that actually leads to your death as a result of being HIV positive. That's why AIDS drugs and the AIDS cocktail treatments are geared towards preventing the virus from replicating itself. Because if you can stop the virus from replicating itself, you can stop the loss of the immune system, and a person can live a normal, healthy life. Anyway, I digress again, but that's a little bit interesting, more interesting information about viruses. So we know that the the T2 bacteriophage has to take over an E. coli cell in order to make more copies of itself. So it has to insert something into the E. coli cell to reprogram that cell into a virus factory. What Hershey and Chase set out to do is to find out whether it was the DNA or the protein of the T2 bacteriophage that was inserted into the cell and reprogrammed it to become a virus factory. What they needed to do, however, was to be able to clearly differentiate between the viral DNA and the viral protein. And they used a very simple observation. DNA contains phosphorus, protein never does. Protein contains sulfur, DNA never does. And there are two radioactive isotopes, phosphorus-32, which is highly radioactive, and sulfur-35, which is nearly as radioactive as phosphorus-32, that can be used to differentially label the viral DNA and the viral proteins respectively. All you need to do is to grow new virus particles in the presence of the appropriate isotope, and all the newly created virus particles as a consequence of the viral infection of E. coli will contain the isotope in the appropriate molecules. So they grew some bacteriophage in the presence of sulfur-35, they grew some others in the presence of phosphorus-32, and labeled the protein and the DNA respectively. Then they transferred those to new solutions containing fresh E. coli, allowed the virus particles to go ahead and infect the E. coli cells, waited a few minutes to let them do that, then put those solutions into a blender and turned the blender on. That whizzed the cells round and round. The bacterial cells remained intact. They're too small and too tough to be broken by the blender, but anything that's stuck on the side of them, that gets sheared off, that gets torn off by the action of the blender. So now you've got these two solutions of infected E. coli cells with something, perhaps, that might have got injected into them by the virus particle. Now you want to find out, is it the DNA or is it the protein? Well, the way to do this is to take those solutions and place them into centrifuges. When you turn the centrifuge on, everything gets spun and an artificial gravity field is created. Let me get the pen out. The artificial gravity field is created that pulls everything to the bottom of the tube. Anything that's large, like a bacterial cell, which is relatively large, gets pulled to the bottom of the centrifuge tube by the artificial gravity field. Anything that's small, like the fragments of the virus that have gotten torn off the outside of the cells by the blender, well, those stay in the solution, what's called the supernatant. So now, having run the centrifuge at the bottom of each tube, you've got this tight little pellet of bacterial cells. In the supernatant, you've got whatever remnants of the virus were on the outside of the cells and got ripped away by the blender. So now all you need to do is to check and find out which of the two isotopes are in the pellet and which of the two isotopes are in the supernatant or the solution. So when they checked the solution, they only ever found the sulfur-35 there. The sulfur-35 never ever got into the pellet, indicating that the viral proteins always remained on the outside of the E. coli cells. They never got inserted into the bacterial cells because the sulfur-35 never got in there. When they went checking for the presence of the phosphorus-32, however, that was always in the bacterial cells. It was never in the supernatant, indicating that the viral DNA entered into the bacterial cells. It didn't remain in the remnants of the virus particles. What that told Hershey and Chase was that it had to be the viral DNA that was reprogramming the E. coli cells and turning them into virus factories. But what was also known was that bacteria themselves, like E. coli, had their own DNA. So what it was telling Hershey and Chase was that the bacteria were ignoring their own DNA and taking notice of the viral DNA, and that's what was reprogramming them. In other words, DNA contained the genetic instructions. It was the molecule of inheritance. And of course, it was well known that all cells contain DNA in their chromosomes, eukaryotic cells and prokaryotic cells. So it stood to reason that if the viral genome, the viral genetic material, was DNA, and by extension, the bacterial DNA must be its genetic material, then by a further extrapolation or extension, DNA in any cell must be serving as the molecule of inheritance. It must contain the genetic instructions. And Hershey and Chase went ahead and published this paper in 1952. And I forget exactly when in 1952, but I believe it was mid to late 1952 that they published this paper. And at this point, everyone reached the same conclusion. DNA must in fact be the molecule of inheritance, not proteins. And immediately Pauling and his group started looking at DNA along with other people. This is when Watson and Crick restarted their look at DNA with the publication of Hershey and Chase's paper. Pauling was considered to be the person who was leading the pack in terms of trying to figure out what the structure of the DNA molecule might be like. And in fact, he used some X-ray crystallography information, the same sort of information that Watson and Crick would get in a less than upfront way from Rosalind Franklin, and more to come, from people that he was collaborating with in the US. But the X-ray crystallography data that he got access to was not the best. And he interpreted the structure of the DNA molecule as being a triple-stranded DNA molecule. And he actually went ahead and published that. And having published that and claiming primacy, the first person to publish, he sort of established a little bit of a, again, a trend of people's thinking, because Watson and Crick, when they first started thinking about the DNA molecule, also thought of it as perhaps being a triple-stranded molecule. It was only when they got access to Franklin's picture, which was by far and away the cleanest picture of DNA, that they started to realize that it had to be a double-stranded molecule. In fact, Franklin had already recognized that it was going to be a double-stranded molecule on the basis of the picture that she had generated. But we're getting ahead of ourselves a little bit here. So people are now convinced that it's DNA that is the molecule of inheritance. So what was actually known about DNA at this point? Well, a lot of the chemistry of the molecule was known. It was known that there were 4 nucleotides. That was something that was known pretty early on. In fact, very early in the 20th century, the basic chemical makeup of DNA was pretty well known. The presence of 4 nucleotides, even the chemical makeup of the 4 nucleotides was pretty well understood. It was known that each nucleotide was built around the framework of a 5-carbon sugar, deoxyribose, and so you could see that here. You'll notice that the carbons in the sugar are given the designation as a number plus a little tick mark. That tick mark is called a prime mark. And so we call the carbons as 1', 2', 3', 4', and 5'. The key numbers are 3' and 5', which hopefully you've already started to figure out. But those are the key numbers because those are the carbons in the sugar ring that are used to form the backbone of the DNA molecule. The nitrogenous bases that identify each of the 4 nucleotides or distinguish each of the 4 nucleotides are in turn attached to the 1' carbon of the deoxyribose. So in case you're curious, why the prime designation? Well, the answer is because there are carbons in each of the nitrogenous bases as well. And so to differentiate between the carbons in the nitrogenous bases and the carbons in the sugars, the organic chemists that figured out these structures decided to use the prime designation for the carbons in the sugar to differentiate them from the carbons in the nitrogenous bases. That's the reason why. So each of the 4 nitrogenous bases is attached to the 1 carbon of the deoxyribose sugar. So what about the 3' and the 5'? Well, on the 3' of the sugar is a hydroxyl group. This is an unmodified group. And so at one end of a polynucleotide strand, a strand of nucleotides linked together, there's going to be what is referred to as the 3' end, where there is a hydroxyl group, and you can see that here. Let me change it over to red so that it's a little bit more outstanding. So here is our 3' with the hydroxyl. This is our 3' end of the polynucleotide strand. The phosphate of each nucleotide is then attached to the 5' carbon, which actually lies outside of the ring because of the way the ring forms. And if you're not sure about how the ring formed, why that should be the case, you can go and review back in chapter 5, although it's not really essential that you understand how the rings form, just that the ring does form. And the 5 carbon sugar lies outside it. That's got the phosphate group attached to it. So at the other end of the sugar, and let me move this for a moment so I can see the other end, you're going to have a phosphate group that is unmodified and not linked to anything other than the 5' carbon. So this is that phosphate group attached to the 5' carbon. And so we refer to this end as the 5' end. So there is an endedness, if that's a word. I suspect it's not, but hey, what does it matter? You can create your own words at times. There is an endedness to the strand. There is a clear 3' end. That's where you've got the hydroxyl group attached to the 3'. And you've got a 5' end. That's where you've got the unmodified phosphate group attached to the 5' carbon of the sugar. And so then you've got this alternating sugar phosphate, sugar phosphate, sugar phosphate that forms the backbone of the polynucleotide, and then the nitrogenous bases point off to the side. And the structure of all the nucleotides, the structure of the nitrogenous bases, the sugar, of course, and the phosphate groups, those are all well known by the time Hershey and Chase are publishing their paper in 1952. This is all well known. So the nucleotides were well known. What else was known was that the composition of a DNA molecule from a particular species had a very unique nucleotide composition that was unique to that species only. And all individuals within a species contained a DNA molecule that had the same nucleotide composition. But when you compared DNA from different species, the nucleotide compositions were different from species to species. But within a species, every individual shared the same base percentage composition for their DNA molecules. And that should have been a big flag to everybody, because that's exactly what you would have expected from the molecule of inheritance. Every species should have had a unique form of that molecule, whatever it is, that was unique to them, and every individual within that species should share that common uniqueness. So what did Erwin Chagall actually publish in 1947? Because he's the individual who figured this out. Well, what he was able to show was, number one, every species had its own unique DNA molecule on the basis of nucleotide composition, and every individual within the species had shared that same nucleotide composition. But what he was also able to show was that the percentage of the A nucleotide pretty much matched the percentage of the T nucleotide in every DNA that he looked at. So if you look at E. coli, wheat, sea urchin, salmon, human, ox, if you know the percentage of A, you pretty much know the percentage of T for all of those. Each one is unique, but the same basic principle applies. A and T are present in essentially the same percentage. Likewise for G and C. The G percentage and the C percentage were essentially the same. And irrespective of whichever DNA you looked at, the same thing. If you knew the G percentage, you pretty much knew the C percentage. Now, we know what that actually results from these days because we know so much about the DNA molecule. But at this point, no one knew what this meant, other than it was something that was consistent in DNA molecules, no matter where you found them. If you knew the percentage of A, you pretty much knew the percentage of T. If you knew the percentage of G, you pretty much knew the percentage of C. And of course, every species has its own unique percentage base composition, and every individual within the species shares that same percentage base composition. These would become known as Chargaff's rules. Now, there's probably a reason why people didn't pay a whole lot of attention to what Chargaff was saying, because it turns out that Chargaff was a pretty difficult individual to work with. And he was pretty dogmatic in terms of the way he presented his information and what he thought. And so I suspect that even though he had some really good data here that would ultimately be very important to Watson and Crick, a lot of people just ignored it because they found Chargaff to be just too difficult to deal with. So in terms of the chemistry of DNA, there was pretty much a lot of information available. But in terms of the physical structure of the DNA molecule, well, there the information was still a bit fuzzy. As I said, the data that Pauling was depending on, coming from some of his collaborators who had started doing X-ray crystallography of DNA, the pictures were very fuzzy and not easy to interpret. And in the case of Pauling, he allowed himself to be led by his understanding of proteins and started to apply what he knew about proteins to the structure of DNA, rather than trying to start with a clean sheet and completely fresh, open ideas about what the molecule should look like. And this, probably more than anything, is the reason why Pauling, as much as clever as he was, and as far ahead as he was initially in figuring out the structure of DNA, would ultimately miss out, because he allowed himself to be led by his own opinions and what he knew about other molecules. And again, it was very difficult to get him to change his mind once he'd made up his mind. He was pretty dogmatic too. He was, in some circumstances, pretty difficult to work with at times. In the UK, Wilkins and Franklin were working at King's College. Now, the relationship between Wilkins and Franklin was, at best, they tolerated each other. And the reason why was because both viewed their relationship, their working relationship, very differently. Wilkins had always understood that Franklin had been employed to work for him at the college, whereas Franklin always understood that she had been hired to collaborate. She had an independent laboratory, but her laboratory and Wilkins' laboratory would collaborate on the work on proteins, which they initially started working on. And that's why they were doing X-ray crystallography. And that understanding of their relationship would be central to how Watson and Crick got access to Franklin's photograph. If in fact Franklin had been working for Wilkins, then any data that she generated would be owned by Wilkins as the leader of the group, and he could decide who to show it to. If, however, they were collaborators as Franklin viewed their relationship, then the data that Franklin generated was her data, and it was up to her to decide who to show it to. And so you can understand why there would be a real degree of tension between the two of them because they really did not see eye to eye about their working relationship. As it would play out, Franklin and one of her graduate students started playing with salmon sperm DNA. Almost immediately, the Hershey Chase picture came out, or the paper came out. And Franklin was extremely skilled at creating crystals of very large molecules. She had, after all, been working with proteins for many years and generating X-ray crystallographs of protein crystals. And she simply applied that skill to generating an X-ray crystallograph of DNA. And so she and one of her graduate students generated this picture that you can see here. And this is a very, very clear picture. You may not think so, but it is a very, very clear picture of the physical structure of the DNA molecule. And it tells you quite a bit about the DNA molecule. Now, once Franklin and her graduate students had this picture, Franklin wrote up a little bit of her thoughts about what the molecule was like. And it's almost certain that within 6 months, if Watson and Crick hadn't beaten her to the punch, she would have written up a paper describing the three-dimensional structure of the DNA molecule, because it's pretty clear what the structure of the DNA molecule is from this. Although Franklin initially misinterpreted the model because she didn't see it as a double helix initially, but she would reach that conclusion pretty quickly. In the meantime, having generated this picture, written up her initial thoughts, she went on vacation. And while she was on vacation, James Watson came down to visit Maurice Wilkins, because Wilkins, Watson, and Crick were pretty friendly with, Watson and Crick were pretty friendly with Wilkins. They also, Watson and Crick also knew Franklin. Crick and his wife were reasonably friendly with Franklin. But there was a lot of animosity between Watson and Crick, also between Watson and Franklin, in a similar fashion as between Franklin and Wilkins. So when Watson came down to visit Wilkins, they were talking, and unbeknownst to Franklin, Watson and Crick were starting to work on the structure of DNA again. And Wilkins knew about Franklin's picture, and he showed it to Watson while Franklin was on vacation. Now, one thing that Watson did have that was to his advantage was a photographic memory. So even though he only saw the photograph for perhaps 30 seconds, maybe a little bit longer than that, he was able, when he got back to Cambridge, to reproduce the picture exactly so that he and Crick could start to try and figure out what the picture was showing them. Now, they were not X-ray crystallographers. They could not figure out everything that the picture was telling them. But there is one clear detail here that even Watson and Crick could figure out, and that was that this is a double helix. The characteristic that tells you that it's a double helix, let's go back, is this, the X. That is a clear signature of a double helix. And while apparently Franklin missed this initially, she would very quickly realize that it was a double helix. She actually provided, through discussion with Watson and Crick, the other pieces of the puzzle that come out of this picture. She did not realize what Watson and Crick were doing because they were keeping that very much to themselves. Even Wilkins didn't know exactly what they were up to. So they kept this all pretty much to themselves. So unbeknownst to Franklin, she basically gave them the other pieces of the puzzle. So what are the other pieces of the puzzle? These two big signals are the sugar phosphate backbones. When Watson and Crick first started building a double helix model, they actually put the backbones in the middle of the molecule, following somewhat along the lines of Pauling's triple helix model, where he had the backbones in the middle, but had 3 backbones in the middle. Watson and Crick put 2 backbones in the middle because they were pretty confident that it was a double helix type molecule. But they put the backbone in the middle with the nitrogenous bases pointing out. Franklin said, no, these 2 signals tell me that the backbones are on the outside, the nitrogenous bases have to be pointing to the inside. Then what she was also able to tell them from these signals is that there are cross-links between the sugar phosphate backbones, and those cross-links must be formed by the nitrogenous bases. And what she was able to determine was that if you look at one turn of the helix, one spiral turn of the helix, there are 10 cross-links per turn. So 10 cross-links per turn of the helix. More than that, she was also able to say, on the basis of these very strong signals, that the molecule was of consistent diameter. It didn't vary in diameter. It was of constant diameter, the full length of the DNA molecule. She could even estimate the distance across from one side of the molecule to the other side of the molecule, and she pegged that at around 2 nanometers. Now, a nanometer is 10 to the minus 9 meters. It's an incredibly small number. And it would turn out that she would be pretty much spot on. She kind of missed on how big a turn was. She knew that there were 10 cross-links per turn. She estimated the distance between each of the cross-links and was able then to estimate how much distance one turn represented. And she was a little bit out on that one. She overestimated the distance between the cross-links a little bit, but she was very close nonetheless. So she had all of this physical data. And as you can see, if she'd been given the opportunity, she probably could have generated a paper describing the structure of the DNA molecule. What she didn't realize was she was giving this information to the 2 guys who were using it to figure out the structure of the DNA molecule, because they'd already seen the picture. They knew a lot more than she realized about the structure of the DNA molecule, and they were actively pursuing it. So Watson and Crick took this information and started building a new model with the nitrogenous bases pointing inwards and the sugar phosphate backbones on the outside. Their initial idea was to form the cross-links by having 2 A nucleotides form a link, 2 G nucleotides form a link, 2 C nucleotides form a link, and 2 T nucleotides form a link. But there's a problem with that, because as you can see from this picture, T and C nucleotides have a single ring structure, but A and G nucleotides have a double ring structure. And what that would mean is that the cross-links formed by 2 double ring structures would be longer than 2 cross-links formed by 2 single ring cross-links or structures. And that would mean that the molecule would be of variable diameter rather than constant diameter, as Franklin indicated was the case. So that meant that the only way to create a molecule of a constant diameter was to have a double ring with a single ring. But which ones to pair up? Well, that's where Chargaff's numbers came in. Remember, if you know the percentage of A, you know the percentage of T. If you know the percentage of G, you know the percentage of C. Well, A and T happen to be a double ring, single ring pair, as are C and G. There's the solution. If in fact A and T always pair up, then you would expect the percentage of A to match the percentage of T. And if C and G always pair up, then you would expect the percentage of C to match the percentage of G. So that must be what the nature of the cross-links are. But then there was still a problem, because they couldn't see how C and G and A and T could necessarily pair up. It puzzled them. And they were discussing this with one of the organic chemists that had a lab down the hallway from theirs in the old Cavendish Laboratory building, which I've also been visited. That's in the heart of Cambridge. And you can go in there and you can see where they did their work. And there's a nice little, there is another museum in there of scientific instruments, and it's quite fascinating to see some of the scientific instruments in there. Anyway, I digress. There is a plaque on the outside of the building that says that this is where Watson and Crick did their work. And oh, by the way, down the end of the road is the Eagle Pub, where Watson and Crick were known to go every Friday afternoon to have a meal and to drink and to think and talk over what they were working on as far as DNA was concerned. And I was hoping that there would be a spot inside the pub that was known as the Watson and Crick table or something like that, so that I could sit there and have lunch where Watson and Crick did their work. But apparently, no, there wasn't a particular table, or if there was, I couldn't see any indication for it. Although there is a sign outside that indicates that this is where Watson and Crick were. So I had lunch in the pub when I was there. Again, I digress. So Watson and Crick are talking to this chemist, and this chemist actually knew something about the structure of the nitrogenous bases. And he said, if you look at these nitrogenous bases, they actually come in two chemical forms. You're actually working with the wrong chemical forms. You need to think about the other chemical forms, because if you look at the other chemical forms, it's really obvious how A and T could pair up, because in that form, it's possible for the A and T to form two hydrogen bonds. And likewise, in that alternative chemical form, the C and G pair would form three hydrogen bonds. But in order for that to happen, they have to be oriented in a very specific way. Because if they're oriented in the wrong orientation, they simply can't form the hydrogen bond. So there's a very specific way in which the C and G must be oriented and the A and T must be oriented in order for them to form the hydrogen bonds that we now know exist between them. And the only way to get them into that right orientation is for the two polynucleotide strands to be anti-parallel to each other. So what does that mean? Well, if they were parallel to each other, the two strands would be 3' to 5' going in the same direction. That's if they were parallel. But we know that that is not the case. They are not parallel to each other. We know that they have to be anti-parallel to each other. So what that means is that one strand goes 3' to 5' in this direction, but the other strand goes 3' to 5' in the opposite direction. That is what is meant by anti-parallel. So Watson and Crick had to build this into their model. And we now know that, in fact, this is absolutely correct. The two strands are anti-parallel to each other. That will have consequences when it comes to replicating the DNA molecule, as we will discover in the next section. So taking all of this information together, Watson and Crick then drew up their double helix model and published the paper in Nature, as I said, in April. It's actually April 25, 1953. In that paper, not only did they describe the DNA molecule, the structure of the DNA molecule, they made two predictions about what these structure would tell you about the function of the DNA molecule. The first was that information would be stored in the DNA molecule by the order of the nucleotides, something that would take about 12 years to figure out in total, by several groups around the world. But the major group that got involved in this was a group led by a researcher named Marshall Nirenberg, located at the NIH labs in Washington, DC. And it would take approximately 12 years, somewhere around 12 years, to figure out all of the coding information that we now recognize as the genetic code. And when we cover chapter 17 in the next presentation, we'll talk about the genetic code and what the information coding units represent and how that information can be interpreted at the cellular level. What they also said in a single sentence in this particular paper, but they actually expanded on it in the paper that they wrote with Wilkins in the same journal, was that it had not escaped, and I'm kind of paraphrasing here, but this is pretty much what they said. It has not escaped our attention that our model also provides an explanation to how the DNA molecule could be replicated. In the second paper that they wrote with Wilkins, they expanded on that and they used the term semi-conservative replication to identify the process by which they believe the DNA molecule would be replicated. Now, in the next section of this presentation, we're going to actually talk about what that means. For now, let's take a break, because we've been going for a while. And look at a couple of concept check questions, and then take a break. And when we come back from the break, I'll give you the answers to the concept check questions. So here are the questions. If you're given a polynucleotide sequence like GAATTC, and this is a known sequence that exists in DNA, it has a special purpose, but we don't need to get into that right now. Explain what you would need to know in order to identify which end is the 5' end. I just need to read what I put there. So if you've got this sequence, what do you need to know in order to be able to identify the 5' end, and I guess by extension, the 3' end as well? Then the second question is, Griffith was very surprised when the mouse injected with the dead S cells and the live R cells died, and he was extremely surprised to get that result. What result do you think he might have expected and why? So I'll see you on the other side of the break with the answers.